ask you questions because the doctors aren't giving them answers. And it's going to be up to you to help explain why you're doing or you're having them do something. So the more you can think about having damage, let's say we damage this, or I don't know, here or wherever, the better your understanding is in order to explain it to somebody else. So, here we go. The tegmentum, the tectum, the basis. The tectum, overwhelmingly, the two caliculi. The tegmentum, lots of nuclei. The reticular formation here is mostly nuclear in this case. There are a few fiber tracts. And then the basis, overwhelmingly fiber tracts. So here was today's figure. All right, this was it. You should have been able to label the superior colliculus, the anterolateral system, any of these cortical tracts, substantia nigra, and the red nucleus. Other than that, anything else is a bonus, so to speak, for you, what you know in there. The more you know in these areas, the better it is. So, we looked at we touched on, we really didn't do much with this, okay? We did a little bit here and there. We're going to come to it. We talked briefly about nucleus solitarius, the two caliculi, the superior olive, and things like that. We'll come to it. Here we are, the superior colliculus. What's coming to it? What's involved? All right. Whenever you have nuclear structures in the brain, there are two sets of issues. Where are signals coming to it from, and where do the signals leave? Where are they going to? So what signals are coming in, and what is the target of the output? And then the question is, what is the result of the pathway? So if you can answer those three questions about any nucleus in the brain, you are ahead of the game. The red nucleus, we have the two portions, the parvocellular, the magnocellular, inferior olive, outputs to the rubrospinal tract. So the targets are slightly different. Substantia nigra is kind of a big one for you. You have to understand substantia nigra. Substantia nigra is very, very intimately associated with Parkinson's. You will have patients with Parkinson's, so just be ready for it. We then started to work our way down from the midbrain through the upper part of the brainstem, headed towards the lower brainstem. So you've got the inferior colliculus here, the anterolaterals, the tectospinal tract, rubrospinal tract. What I said you should be able to label on something like that. So today I gave you a figure with lots of lines pointing in. I'm not going to get rid of all of those lines. You're going to have lots of lines pointing at figures if I use any of these figures. And it's going to be up to you to realize, oh, I'm only supposed to know so many of these things. So don't try to learn this up here. Learn it here. Learn this over here. Do you see what I'm saying? Learn the actual structure in the brain instead of the arrow that leads to it. Yeah. So we still have the rubrospinal tract in here. It's just flattened out. As we descend into the lower part of the medulla here, we have all of these important nuclei. So we have nucleus solitarius, or solitary nucleus, nucleus ambiguus, the inferior olive in here. So this, in its entirety, is pretty much the inferior olive. As I said, I don't expect you to be able to label the individual components in there. But I could ask you a question, and I could say, okay, so on here, got this figure, okay? Show me which one of these nuclei are involved with cranial nerve 10. Show me which one's involved with cranial nerve 9. And you go, oh crap, I didn't learn it that way. Well, you should. These two, solitarious and ambiguous, are highly associated with cranial nerves. So, got to work on that. Oh, look at this. This kind of summarizes a lot of what's going on. So we've got the 4 nuclei here, and these are all what? Oh, look at this. These are all parasympathetic functions, mostly, in here. So we go down to just about the most inferior part of the medulla. So we're just superior to the spinal cord here. We've got the pyramids in here. We've got the decussation sitting back here. We've got the inferior olive, once again, sitting over here. And then we have the gracile nucleus and fasciculus. We have the cuneate nucleus and fasciculus. Those should be identifiable for you. This is literally as deep as it goes. I mean, if we were to take a slice below this, we would pretty much be in the spinal cord. The pyramids, and all of these tracks piled in over here. Once again, we've got all of the tracks. This makes perfect sense. It should make perfect sense to you. If this is just superior to the cord, all of these tracks make up the cord. So now what's happening is you've got these fiber tracks coming up from the cord, like a whole bunch of fingers. But they're encountering these nuclear areas, these areas where you have nuclei. So the tracks are coming up and they're kind of surrounding them and interweaving with them. And so they're going to be kind of coming up and doing things like this. Well, obviously in the pyramid they are going to be doing things like that. But the tracks themselves are coming up, and we're going to see, you notice in here, most of this is dark. Why is it dark? It's a myelin stain, so it's myelinated. But what you see are some very light areas as well. Those light areas are packed with cell bodies. The dark areas are fiber tracks. So please be aware. I'm in the process of losing my voice, which is probably going to happen by the time your exam comes along. Pontomesencephalic reticular formation. Okay, so we have the pons here, we have the medulla here. This is the mesencephalon, which is why we have that name. And we have inputs coming from all over the place to this region. Your reticular formation is critical to understand. Reticular formation, remember, is highly associated with your various levels of consciousness. And then we had projection systems. Some of these are going to be in the reticular formation, some of them are going to be in other midbrain regions. So with the cholinergic projections, what's the neurotransmitter? Acetylcholine. Cool. We have this basal forebrain area. Okay, it's at the base of the forebrain. Nucleus basalis is buried in here. All right. This fiber tracks lead to the entire cortex. That is the cholinergic fiber tracts in the cortex, for the most part, originate in nucleus basalis, the area of nucleus basalis. But then sitting right deep in here, okay, so in this point right there, we're actually in the upper part of the reticular formation. We have that pedunculopontine nucleus. Oh my God, yeah, all right? It's a pontine nucleus, basically, okay? Peduncle. Does that mean anything to you? It should. It's located adjacent to the peduncles, coming from the cerebellum to the pons. From there, we stimulate the whole midbrain, the epithalamus, the thalamus, less so to the anterior midbrain. We stimulate the cerebellum, the pons, and the medulla. By stimulate, I mean we have stimuli. Is it an excitatory or inhibitory stimulus? It's excitatory. Acetylcholine is excitatory. At this point in time, you should review whether neurotransmitters are excitatory or inhibitory. It's just important to know. We looked at dopaminergic projection systems here. We're going to project to the striatum, which is the major part of the basal nuclei or basal ganglia. We're going to project to the limbic system, and we're going to project to the forebrain. These are three different projection pathways. Some of these projections come from both substantia nigra and the ventral tegmentum. Some come only from the ventral tegmentum, and some come only from the substantia nigra. You should know which is which in there, because it's important. And what this shows you is why with most of the progressive movement disorders, so things like Huntington's and Parkinson's and such, we involve movement, spastic movements initially, that can sometimes lead to terminal loss of movement. By terminal, I mean loss of movement that's going to lead to your death. We see that. We see functional problems with the limbic system leading to inappropriate behaviors and memory function problems. We see prefrontal cortex. You lose your executive suite of decision making and all sorts of things. So it's not surprising, because these are all dopaminergic pathways. We looked at noradrenergic. This is coming from a wide variety of cells in the nuclei, in the reticular formation. And what is it affecting? Oh, medulla, the spinal cord, the pons, the cerebellum, the thalamus, and the cortex. So basically it affects everything. So, but you should be able to look at, and tell me what's going on here. Locus coeruleus is involved with this. We've touched on locus coeruleus a few times. Yeah?
When we are asked questions about this, are we going to have images to maybe help us?
I would, if I'm going to ask you something, what I would do, and I have the ability to do this, is give you this brain without all of the red stuff on here, okay? And I might say, show me the projection system for this neurotransmitter. Is that reasonable? The answer to that is yes.
Is it that the projection systems are in the reticular system?
In the case of noradrenergic projection system, the cell bodies are in the reticular formation.
Oh.
This is the reticular formation right here. This just about defines the reticular formation.
When you say reticular formation, is this coming from the very
No.
top? Or
No, the reticular formation, you remember, is a whole series of structures right here.
Oh,
That's the reticular formation.
this part.
Yeah, it's not the entire